Hi, today we're picking up on where we left off, move that out the way, with the Neoproterozoic era. And this is going to be from about 1 billion years ago to about 541 million years ago. This is going to highlight a significant period in the adaptive radiation of animals. It's going to begin with sponges, which are going to start to appear around 890 million years ago. So these are called choanoflagellates. They're going to extend a group of colony-forming flagellated protists that were believed to resemble a common ancestor of animals. So this is an idea that's going to be based on morphological and physiological similarities of cells with the sponges, which are going to be the choanocytes. The idea of evolutionary link between the choanoflagellates and the choanocytes was first going to be discerned in 1841 by a French biologist, Félix Desjardins, and this is going to be validated through molecular evidence. So here we're going to look at these different pictures that we have here, which are going to lead to the development of when we first start to see sponges. So we have the choanoflagellate on the far left-hand side. It changes over into the choanocyte. Cyte, remember, means cell, so this is going to be a cell that resembles the choanoflagellate, and then those are going to resemble what we find within the sponges. So earliest record of animals is going to show that they are going to correlate with the fossils that were discovered in the Ediacara Hills of South Australia, which is going to be dated around 560 million years ago, and this is going to represent the soft body relatives of sponges that are going to start to develop. So sponges, cnidarians, and the mollusks. Then we're going to head into what's known as the Cambrian explosion. Cambrian, of course, we're going to be talking about the skull, the head, the brain area. And so this is going to be from around 535 to 525 million years ago. During this time, we're going to have rapid diversification events occurring, which are going to be signified by numerous fossils that display diverse bodies. And with those, we're going to have a major evolutionary innovation that was believed to coincide with the close relatives of the extant animal phyla. Extinct means it's no longer here, but if it's extant, that means that it is still living. So you do need to know the difference between those two terms. So many fossils of the time period are going to be discovered in what's called the Burgess Shale, and that's going to be an outcropping that's located in Yoho National Park of British Columbia, and it was discovered by American paleontologist Charles D. Walcott in 1909. And so you can see some of these rock formations and discoveries at the bottom of the slide here. Now with this Cambrian explosion, there's going to be several different hypotheses. First, we have the Neoproterozoic. This is going to be the oxygenation event that occurs during the Precambrian-Cambrian transition. So we're looking at about 850 to 450 million years ago. It was facilitated to support and survival of large animals whose higher metabolisms are going to be supplemented by elevated levels of atmospheric oxygen. Remember that oxygen is going to be needed for cellular respiration, so in order to get energy. Evolution of dynamic predator-prey interactions is also going to be a second event. That's exhibited a strong influence of evolution of renewed predatory modes, including the use of things such as sharp claws and jaws that are going to complement the larger diverse body forms, and effectively it's going to result in the differentiation of specialized tissues and organ systems for these animals to help put them on top. Countering the strong effect of natural selection, it's going to be favoring predation. So we have prey species that are going to start to evolve, and they're going to actually have different defensive innovations that are going to come about because of the predators becoming so much more equipped. Things like this are going to be mineralized skeletal structures, so we're going to start to see things like spines and spicules that are going to come about, and that's going to help with protection of these animals. We start to see shells coming about. Of course, that's going to help with protection, and armor to minimize their vulnerability to these predators. I have to stop it for a minute. All right. So the third thing that's going to come about that is going to be a major event is the appearance of the Hox or homeotic genes. These were first determined or identified by English biologist William Bateson in 1894 and later investigated by the American scientist Thomas Hunt Morgan. Scientists Edward B. Lewis, Christian Nusland Volhard, and Eric F. Weishaupt is going to be the first to discover and identify these genes that are associated with coordinated segmented parts of a body, and they're going to find this in larvae of Drosophila, and Drosophila are going to be fruit flies, and this was done in the 1880s. They find out during these studies that they're going to be responsible for variations in differentiation and specialization of tissues, of organs, and organ systems in animal groups, and so they're able to carry these and follow these throughout different types of organs using these same genes. So here's an example of the genes that they found and how they correlate with the Drosophila or the fruit fly. Now, major evolutionary events with the introduction of the vertebral column are going to come about. That's going to be a mineralized endoskeleton that's going to start to form a bone or cartilage in a centralized nervous system with a complex brain. So before, we did not have this. It's going to be prompted by a whole genome duplication. That's going to be a phenomenon that results in multiple copies of a gene, including those Hox genes that we just talked about. One set of Hox genes is going to be found in invertebrates, 4 sets in vertebrates, and so make sure you remember that. Earliest fossils are going to be recorded of these transitions, and it's going to occur around 540 million years ago when this starts to appear. And this is going to be our ancestral chordates, or the first chordates that come about. Now, chordates are going to share several morphological characteristics. They're going to have different hallmarks, including things like a notochord. They have gill slits at some point in their life, and a dorsal hollow nerve cord, which is going to set them apart from other phyla in the animal kingdom. Hemichordates are going to be considered the best extant example of this evolutionary transition because they are going to be descendants of ancestral chordates, and they demonstrate invertebrate and chordate features, so they'll have both of those. Ostracoderms are going to be appearing around 530 million years ago in the Cambrian period. They're going to be small, jawless, and finless fish that are going to be encased in a bony dermal armor. These are going to give rise to things like hagfish and lampreys before the extinction of the Denovian period. The placoderms are going to be heavily armored fish with jaws and fins that are going to be able to enable them to dominate the aquatic environment during the Silurian period, which is going to be around 444 to 419 million years ago, and the Denovian period, which is around 419 to 359 million years ago. These are going to be considered the predecessors of modern-day fishes. Now we're going to start to see those fins and jaws come about. Water-to-land transition and early tetrapods are coming up next. So after that happens, they're going to take the place at the end of the Denovian periods, the end about 385 to 360 million years ago. This is going to include the evolution of walking limbs. With that, now that they're coming out of the water, they're going to have more atmospheric pressure on them, so they're going to have denser skeletons to be able to withstand gravity. Also, they have been used to being able to breathe in water. Now they need to have efficient lungs. They have to be able to breathe this atmospheric oxygen. And with that, they're going to need a better circulatory system because beforehand, remember, they were using osmotic pressure and hydrostatic pressure, and so they were using water as it flows through them as they were swimming. So scientists are going to suggest this major step in evolutionary history of vertebrates is going to involve ancestral lungfish moving between water sources and on land. So diversification of tetrapods during the Carboniferous period, that's around 359 to 299 million years ago, is going to be the result of early animals that are going to start to utilize the abundance of resources and protection available that they have now on land. Fossil records are going to reveal several transitional forms that have helped scientists better understand the evolutionary transition of vertebrates from water to land using the tiktaalik. And so there's lots of videos, National Geographic, things like that are on the tiktaaliks. All right, next we're going to have movement onto land, exposing early tetrapods to air. Like I said, now that they're exposed to air, they have to worry about drying out. They have to worry about the atmospheric pressure coming down on their bodies, so they have to have tougher skin, tougher bodies. So now that they're exposed to the outer elements, temperature is going to be another thing. So they don't want to have to worry about desiccation or drying out or hot temperatures. Now they're going to have thick, scaly skin. They're going to have internalized fertilization. And we see the appearance of the amniotic egg, which is going to allow ancestral reptiles to be fully adapted to land now because they don't have to worry about desiccation of their eggs. The amnion is going to help to protect the embryo within the watery fluid. We have alanatois, which is going to facilitate gas exchange and removal of waste. We have the yolk sac, which is going to supply food developing for that embryo. And we have the chorion, that's going to provide further protection for that embryo. Samuria is going to be believed to represent a transitional fossil from ancestral amphibians to early reptiles. The evolutionary link between dinosaurs and birds is going to be evident in this transitional fossil that you're going to see here. It's going to exhibit characteristics of both reptiles and birds. So extant birds are going to diverge from dinosaurs about 150 million years ago and still express things like reptilian ancestors had, such as scales sometimes on their legs. We see clawed toes still, and sometimes they will lack teeth though. Sometimes they might have like little serrated edges on their beaks, but they don't actually have teeth now. So they do lack the teeth, but they do still have feathers. Okay, mammals are going to evolve from mammal-like reptiles that are going to appear alongside the dinosaurs about 250 million years ago. Diversification of mammalian species with the typical characteristics that define these vertebrates, like hair, mammary glands. These all are going to be things that are starting to occur or appear between 208 and 178 million years ago. Evolutionary innovations of mammals are going to include things like variable modes of transportation. So now we see things such as gliding, climbing, swimming, and burrowing. Distinct heterotrophic diets, it means they eat other things. So we see them either eating plants for herbivory or carnivory. They're starting to eat other animals. A variety of complex teeth. So we are starting to see incisors, canines, molars, and premolars coming about. And early mammalian species are going to undergo adaptive radiation and begin to occupy the habitats of extinct dinosaurs around 66 million years ago. Now going back to Linnaeus, because we always go back to Linnaeus. Remember, he created the Systema Natura, which is going to offer systemic classifications of animals. It's going to be divided into 6 different classes. We're going to have what's called the Quadrupeda. These are going to be animals or mammals. We have the aves, which are the birds, the amphibia, which are going to consist of your amphibians and reptiles, the pisces, which are fish, insecta, which are insects, and the vermes. Although the term translates to worms, the class is going to include all invertebrates and some insects, or a part of insects, sorry, so not with the insects. So originally, these are going to be classified, the sponges that we come across first, as Cryptogamia. However, discussion regarding groups of different animals is going to prompt him to elevate their status to the animal kingdom in 1767. And so here's Carolus Linnaeus's different animal descriptions and how he separated and classified them, if you want to stop and look over these closer. Jean-Baptiste Lamarck is going to come about in 1793 to 1809. He expands Linnaeus's classification system and introduces 3 new phyla. He introduces the mollusks, the echinoderms, and the polyps. These represent sponges, corals, and the jellies. These phyla, along with insecta and vermes, now are going to constitute the invertebrates. So that's going to be a term that's proposed by Lamarck to differentiate from the higher forms of animals, which are going to be known as the vertebrates, or those with a spinal column and spinal cords. They're going to revise this classification system of invertebrates again in 1801 and include the crustacea. Those are going to be things such as crabs, shrimps, and lobsters, and the aconids, which are going to be spiders and scorpions. We refer to them as arachnids today. These are going to be 2 new phyla that they're going to include. They're going to make another significant change in classification systems of invertebrates in 1809, and then they start to include single-celled animals like infusoires. Radially symmetrical animals like jellies and sea stars are now going to be called the radiaris, barnacles, and they're going to subdivide worms based on their level of anatomical complexity into two separate phyla, the annelids or the verse. And so here is Jean-Baptiste Lamarck's different classification systems that he's going to use in order to develop his different phyla, so you can look at those. Next, we're heading on to Georges Cuvier, 1795 to 1817. He's also going to expand on Linnaeus' early classification system by dividing Insecta and Vermes into 6 separate classes of invertebrates based on anatomical function this time. Later, he's going to modify his system in 1817 and name 4 embranchments of the animal kingdom, which include the vertebrates, mollusks, articules, and rayons. And so with these, they're going to include things like sponges, worms, echinoderms, and cnidarians, and with that last group. Highlights of classification system are going to include delineation of mollusks and cephalopods, gastropods, and acephals. So cephalopods, think of head, foot. Gastropods, think of stomach, foot. And so these divisions of universally known classes of mollusks today are going to be our cephalopoda, gastropoda, and bivalvia. And so that's going to be the last one. And so that means it's going to be a 2-shell system or have 2 valves, so it can kind of open up like that. So you're thinking of things like clams and things like that that fall into that group. So the classification system is going to remain well established, and it's going to be observed by most naturalists until the 1860s. And so here is going to be Cuvier's classifications. Now going into Ernest Haeckel in 1866, he's going to propose the first genealogical classification system. He coins the term metazoa, and that represents multicellular animals with tissues. And the phylum is going to represent the highest categories of the animal kingdom. Classification systems are going to include 4 invertebrate phyla, including the Coelentria, or the jellies, hydras, and sea anemones, the Echinodermata, these are going to be your sea stars and sea cucumbers, the Articulata, or arthropods and worms, and the mollusca. Although sponges are going to be a group that's recognized as animals since 1767, Porphyria is going to be introduced by Robert Grant in 1835, and then Haeckel's going to relegate the sponges to his newly created category kingdom of Protista. So he actually puts them in with the protist in 1866. And he's going to do this because of some observable similarities between their cells and those of protists. And so here is Haeckel's. Now with him, he's going to remove the spongae from his newly created kingdom Protista in the 1870s when he reclassifies the sponges alongside with corals and jellies within the Coelenterata. The placement is going to remain there until William Johnson Solis is proposing the subkingdom Parazoa to represent the sponges in 1885. Here, they do this because sponges are going to lack what they call true tissues from the metazoans. And the true tissues are basically going to be tissues that are specialized to do specific things. Most of the tissues within sponges all do the same thing. With the cnidarians are going to, let me move myself out the way. Cnidarians are going to be the jellies, the sea anemones, the hydras, and the corals. Berthold Hatschik divided the Coelenterata in 1888 into the Spongiaria, the Cnidaria, and the Ctenophora. By the 1940s, preference for the name Cnidaria to represent jellies, hydras, and sea anemones is going to be advocated by Libby Hyman. The name is not going to be implemented permanently into classification system, though, until the 1980s. So the platyhelminths are going to be your flatworms. These first started out as Haeckel's Articulata phylum, which is going to include the Vermes and the division including the platyhelminths, the nemahelminths, and the annelids. Now, the platylemia is going to be established in 1851 by Carl Vogt. Carl Gegenberger is going to be credited for originally naming the worms the platyhelminths and reclassifying them into 4 groups. and you see the 4 groups there in 1859. The Nemertina are going to be removed in 1876, and remaining groups represent the phylum Platyhelminths today. The Nematoda, or the nematodes, these are going to be your roundworms. These are going to go through several different names and occupation of ranks, such as phylum, class, order, and family, to represent the nematodes beginning around 1808, and you can see how it changed throughout up until 1997. Eventually, it's universally recognized by the phylum of scientists, the nematodes, or nematoda, by 1997, and that's still what we go by today. Annelidia, or what we call the annelids today. Lamarck is going to be credited for dividing his original classification of the verse, remember those are the worms, into 2 divisions, one of which is going to be named verse exteriors, a group that includes earthworms, polychaetes, and leeches. In 1809, it's introduced the name annelids to categorize earthworms and polychaetes, while the leeches are going to be sangsu. They remained outside of the group. They're going to make modifications again to his classification system in 1818 with 3 proposed subgroups and leeches occupying their own groups. Georges Cuvier is going to be first classification system that's going to reveal that the original proposal of annelids, but like Lamarck, he's going to introduce 3 subgroups in 1812. Next we have Grubb in 1850, who introduces the Oligochaetia and the Polychaetia. Now with the Annelidia, let me move my face again. Jean-Louis Armand de Quatrefages in 1866 is going to designate divisions among Polychaeta with the Aurantia, free-moving, or Sedentara, which is going to be tube-dwelling. These divisions are going to be followed until the use was going to be abandoned in the 1960s, but terms remain customary in textbooks until 1997 and are still observed in some present-day classification systems. Wilhelm Michelson in 1919 is going to introduce the Cladellata based on presence of the clitellum in earthworms and leeches. Now this term is going to represent the monophyly of the Oligochaetia and the Hirundinea. So we have the Polychaetia and Cladellata, which are going to be this group that represents 2 classes of the phylum Annelid. Moving on to the mollusca. These are your mollusks. These are first separated from Linnaeus's vermes by Lamarck and Cuvier, both who are going to introduce them as a phylum under the name mollusks. Lamarck's mollusks are going to consist of 2 divisions. We have the cephals, or the octopi, or snails, and we have the acephals, which are going to be the bivalves, the conchs, and the whelks. Cuvier's mollusks are going to consist of 6 divisions, and they're going to include the cephalopods, the gastropods, the acephals, and remain in effect until the 1830s. Thompson and Brewster in the 1830s are going to remove barnacles because the scientists are going to discover that their larval stages are going to be like the crustaceans. Then we have Alexander Walowiecki. Then 1866, they removed the tunicates after he observed the presence of the notochord. And remember that notochord is going to be important. This structure is going to be shared with vertebrates during embryonic development. So that's going to move that group. Modern classification system of mollusks is going to resemble Cuvier's and is going to have 3 distinguishable classes. So we have the bivalvia, or the bivalves, the gastropoda, or the gastropods, and the cephalopoda, or cephalopods. The arthropoda, or arthropods, are going to be in Linnaeus's Insecta, and they consist of 7 classes, including the last designated for wingless insects. So we have crustaceans, arachnids, and myropods. These are going to be your centipedes and your millipedes. Lamarck and Cuvier are going to treat Linnaeus's Insecta similarly to revealing classification systems that demonstrate a significant reduction of the grouping. And so they're going to put them into 3 groups, the Crustacea, the Arachnida, and the Insecta. Cuvier's classification of centipedes and millipedes within the division Myropoda And Pierre Latreille in 1825 is going to introduce the use of hexapoda. That's going to represent insects. Lamarck originally proposed the term 20 years earlier, so he just brings that term back. Ernst von Siebold in 1848 is going to be credited with grouping insects, crustaceans, arachnids, and the myriapods under the name Arthropoda upon removal of the annelids from Cuvier's Articulata. Now, Haeckel in 1866 is in his genealogical classification system, he's going to regulate the Arthropoda as a subphylum, further dividing it into gill-breathing arthropods, which are going to be your crustaceans, the air-breathing arthropods, which are going to be your tracheata, and that's going to be named after the trachea, like our windpipe that we have. Reginald Pocock in 1893 is going to remove arachnida from the tracheata and redefine it to include the myriapoda based on the phylogenetic relationships that he believed more closely were exhibited between the two groups. Now they're going to have debates regarding these phylogenetic relationships, and it continues for nearly a century until molecular data establishes a continuity and helps to support the monophyly among these different groups. So we have Arthropoda is going to be a subphyla of the Chelicerata, the Mandibulata, and with that we're going to divide those up even more. Phylogenetic instability is going to remain with many of the arachnids and crustaceans. Hold on. Sorry, I'm trying to keep my computer from going to sleep. I need to use it in a minute. And so with this, Arthropoda is going to delineate specifications that are going to differentiate the crustaceans, the myriapods, the arachnids, and the insects. The Echinodermata. Echinoderms are going to occupy Lamarck's group, a grouping that's going to be separated from the animals of Linnaeus's Vermes. Introduction of Echinodermata is going to be a phylum that is credited to Lamarck. Cuvier's division of echinoderms in 1817 is going to be like Lamarck's, but they're going to be a non-distinct phylum. Then we're going to have in 1854, it's established into Echinodermata, and they're going to use that as a distinct phylum. And then in 1866, the preserved designation of the phylum in his genealogical classification system is still going to be used. Now, echinoderms are going to occupy Lamarck's grouping into the late 1800s and even into the 20th century. So for a century, other important scientists are going to be responsible for shaping the current classification system of the echinoderms. So we have people like Ludwig, Cugnot, and Hyman, which are going to come in and help to shape this. In the early 2000s, the implementations of molecular data into systemics is going to prompt additional revisions of the classification system of the echinoderms, which today is going to be represented by 5 different classes. So we have our sea stars, we're going to have what's called our brittle stars, we have sea urchins, we're going to have sea cucumbers, and then we have the sea lilies. Now moving into the vertebrates. This is going to include mammals, amphibians, reptiles, birds, and fishes. We have John E. Gray in 1825 that's going to make the most significant change to Linnaeus's classification of vertebrates using amphibia to represent the caecilians, the salamanders, and the anurin, or frogs and toads. We have other scientists which are going to come in 1866. That's Kowalski, and they discovered the notochord during embryonic development. Haeckel in 1866 is going to propose the Cordonia as a phylum for tunicates, lancelets, and vertebrates to highlight importance of notochords and their phylogenetic relationship. Lancaster in 1877 is going to be credited for conceiving the modern-day phylum Chordata. That's a term that was introduced by Francis Balfour, which includes the Cephalochordata, the Urochordata, and the Vertebrata. So looking at current phylogenetic classification of animals is basically going to be based on information that's acquired through molecular data analysis. So we're going in, we're looking at the DNA, we're running the DNA, and we're looking at how it matches up with all the other chordates. This information is going to help scientists to compose phylogenetic trees that are represented by several branches starting from ancestral colonial protist ancestors. So the branches are going to split off and include things like protozoa. They're going to have the emetozoa. These are going to then branch off, and then we have what's called the radiata. Those are radial symmetrical animals. We're going to come in and have the cnidaria, and then we have the bilateria. The bilateria is going to be further divided into 3 branches that you see here. And so this is going to be your phylogenetic classification tree or your system that you can follow along. So that's going to finish us up for Unit 18. I will meet you back for Chapter 19.